Biology, the last video. So, where we left off was chapter 21, and I ended up splitting chapter 21 into two separate videos instead of one video, and that made it so that the first half was last week and the second half was the beginning of this week. So this week was originally going to be chapters 22 and 23, but because of bumping the second half of chapter 21 to this week, I am omitting chapter 22 and we're jumping straight to chapter 23, and this is where we will wrap up. So chapter 22 would have covered the circulatory system, so we would have looked at how the heart works and like the blood vessels and how the blood gets oxygen from the lungs, stuff like that. And then also covered the excretory system. With oh no, first the after that after the circulatory, the lymphatic system, which you have these lymph vessels and lymph nodes all throughout your body, and they help filter stuff out of your bloodstream, and they play an important role of helping you get better when you have gotten an infection. And then the excretory system, which is mainly the kidneys, but then also your urinary system. So that's what we're skipping. And then we're doing chapter 23 here on the nervous system. Then chapter 24 is mainly let's see, the endocrine system, which is like your all the different hormones that affect how different aspects of your body work. And then chapter 25, the reproductive system. So we're not quite getting to all the body systems, but we're hitting quite a few. And I thought that you would find the nervous system pretty interesting. So that's why I wanted to make sure that we got this chapter in. So here we go. First, we look at some of the main aspects of the structure of the nervous system. This one, I'm going to do kind of a quick looking at the text and stuff, but let some of the embedded videos do a lot of the instruction, a lot of the talking for me. But looking first at the anatomy of a neuron, there's a cell body. So, like this here would be a cell body, and then this here would be the cell body of a different neuron. And it contains the nucleus and organelles and may receive impulses. Then the dendrites. These are dendrites, these like tree like things sticking out from the cell body. And they are multi branched fibers that project from a neuron. And they receive and relay nerve impulses toward the cell body. So the dendrites receive signals, whether it be receive signals from its surroundings or receive signals from another neuron. And then there are these axon fibers. That's like this axon there, that long. This case looks like a link of sausages. Axon fibers or axons. These are fibers that carry impulses away from the cell body. So the dendrites receive, then the cell body passes along to the axon. The axon transmits the signal away from the cell body. And different parts of the cell body, I'm not going to get into those other than maybe the synapse and synaptic cleft. So at the end of the axon, you have what are called the synapses, and these are the junctions between two neurons. And the synaptic cleft would be the space. It's a teeny tiny space. The video mentions about how big, but a teeny tiny space where the end of one neuron kind of connects. So again, it's a gap to the beginning of another neuron. Okay. All right, and then types of neurons, you have sensory neurons, they receive impulses. You have relay neurons, or the text here called them interneurons, and then motor neurons, where those take an impulse from your nerve, from like your brain, and send it out to a muscle. Okay? And neuron functions, so this is how they pass a signal along. There's something called a nerve impulse, and it's an electrochemical pulse that moves along the membrane of a neuron. So, like this action potential, action potential, action potential. See how it's moving from the beginning to the middle to the end. That would be like the impulse that is traveling down the axon. And the resting potential here is a difference, the relative charge differential between the inside and outside of the neuron. So usually the neuron pumps ions in such a way that it maintains a slight positive charge outside and slight negative charge inside. But then this action potential, it triggers. So when the dendrites receive an impulse and then the cell body transmits the impulse down the axon, that triggers some opening of like transport proteins that allows some of the positives to flow in, and that leaves a slight negative, and that triggers the different parts next to it to do the same thing, and that triggers the part next to it to do the same thing, and it creates this super quick movement of the changes in the action potential, or sorry, in the changes in the potential. What am I trying to say? It's the changing in the resting potential. The action potential is kind of causing that makes the action potential travel. Even if that last wording was slightly confusing, I think you get the gist of what I'm saying. And then there are neurotransmitters. Those aren't shown here, but these are chemicals that are released at the axon terminal, so at the end of the axon, and they stimulate the receptor proteins in the membrane of the next neuron. So as the term implies, neurotransmitters, they transmit the neuron's signal from one neuron to the next neuron by traveling across the synaptic cleft. And there's a video that goes into that a little bit. So speaking of videos, we have a few that give a little bit more of a overview of what we were just looking at, and we'll move on from there.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with the receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
Okay, next.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter. Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission.
Yeah, I thought that might have been the same one again. I don't like how the display thing with PowerPoint works with videos where the video ends and you click the button that always takes you to the next slide, but it just restarts the current slide. Okay, here we go. There we go. Divisions of the nervous system. So the nervous system roughly breaks into the central nervous system and the peripheral nervous system. Central nervous system is basically your brain and spine is the main part of it. And the central nervous system is covered with some layers called meninges. These you maybe have heard of meningitis. That is an inflammation of the meninges. The itis ending means an inflammation of. And yeah, these are membranous coverings of the central nervous system, CNS. And there are three types: the dura mater, the arachnoid membrane, and the pia mater. I'm not going to take time to go over those right now. And then there's a cerebrospinal fluid, and it basically helps to nourish and protect the different parts of the central nervous system. And then the brain. There's the cerebrum that has the left and right hemispheres, the cerebral cortex. I'm going to briefly look at these just because it gives you a little bit of an idea of how many different things the brain does for us. Cerebral cortex. This is the outer gray area. This is like outer gray matter. This is the outer area of the main thing that you think of when you think of your brain. And it's generally responsible for conscious activities. So you decide to say something. You decide to wiggle your finger. You move your arm to catch an object. All that stuff like that. Thalamus. This receives general sensations and quickly decides which impulses are important enough to be relayed to the parietal lobe. So it's both while you're awake and while you're asleep, it's processing the sensory information that it's receiving and being like, let's see, do I want to make him is this important enough to like make him consciously aware of it? Yes, okay. Or no, I don't think so. And I think it's the thalamus doing the work when, like, for example, in college, I could use an alarm to wake me up. And then there maybe is a day where I didn't have to get up as early, and my roommate asked if he could borrow my alarm. And he was getting up around the same time I usually would. But because my alarm was going off for him rather than for me, I knew that going into the night before when I went to sleep. And so it's like my thalamus, when it heard the alarm going off, was like, this is not important information because this is not the alarm for him to wake up to. And so I slept through the alarm. Stuff like that. Then, so let's see, we just talked about the thalamus. So now the hypothalamus. This controls many involuntary activities such as regulation of body temperature. Aren't you glad you don't have to think about raising or lowering your body temperature to maintain a healthy balance there? Or your blood volume. We don't have to think about, I need a little bit more blood in my body. Or the different fluid balances in your body. Then there's the midbrain. It's located between the thalamus and something called the pons, and it contains the centers for controlling body movements and posture. So it like helps coordinate all the different muscles that help maintain our posture that we don't really have to think about a lot of times. Pons. It's almost entirely made up of what's called white matter instead of gray matter, but we're not going to get into that. And it carries information from one side of the brain to the other. There's also something called the corpus callosum that helps transmit information from one side to the other. And that's where you get things like crossing over, or synesthesia is something that happens sometimes, where things like people end up with certain colors or smells or textures associated with numbers and different things like that because they have some additional crossing over between things like the numeric part of their brain and the color or texture part of their brain, the more so than normal. That was the pons. The medulla oblongata. Makes me think of the Ben Stiller, no, not Ben Stiller, Adam Sandler movie, the Water Boy, as the medulla oblongata kind of plays a key role in a couple scenes. Anyway, this is continuous with the spinal cord, and it functions primarily as the relay center between the spinal cord and the brain. And it helps control things like respiration, so your breathing, so you don't have to think about that, your blood vessel diameter, because if you need increased blood flow to parts of your body has to increase the diameter of those blood vessels, and then also increase your heart rate at times. And so all of that kind of stuff is controlled by your medulla oblongata. And then the cerebellum. This monitors and adjusts body activities that are stimulated by other brain regions. And this is subconscious stuff. So, all other stuff that is just going on in the background without you consciously realizing it or having to think about it. And then the other part of the central nervous system is the spinal cord. And there's the peripheral nervous system. And it has three main parts. There's the motor division. This carries information from the central nervous system to the rest of the body, allowing the body to react to the information received through the sensory system. So, like, sensory system comes into the brain, your brain processes it. Sometimes you're aware of it, and sometimes you can decide what to do about it. And then, whether it's you deciding or just your brain knowing what to do, then there's information that goes out to the motor division or through the motor division that triggers different muscular responses in the body based on the information that the brain received. Then there are cranial nerves. These are 12 pairs of peripheral nerves that originate directly from the brain. Most of these control the sensations and movements of the head and neck. And then there are spinal nerves. These are 31 pairs that should say originate in the spinal cord and branch out to both sides of the body. So you have a rough breakdown here. You have the yellow, that's the central nervous system, the CNS, and then all the blue ones are the peripheral nervous system. So yeah, a lot of nerves. And that's not even getting into like the teeny tiny nerves with the sensory nerve endings and yep. Okay, one or two more of these 2 minute, and then we have some other kind of videos that we'll watch.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the divisions of the nervous system. There are two major divisions of the nervous system. The first is the central nervous system, which is made up of the brain and the spinal cord. The second is the peripheral nervous system, which consists of nerves that run throughout the body. The peripheral nervous system itself is made up of two subdivisions. The first is the somatic nervous system, which contains nerves that carry sensory signals from the body to the central nervous system and nerves that carry motor signals from the central nervous system to the skeletal muscles. The somatic nervous system is associated with voluntary movement. When you clicked on this video to play it, the signal to depress your finger was sent from your brain to your finger via the somatic nervous system. The second division of the peripheral nervous system is the autonomic nervous system. The autonomic nervous system is sometimes called the involuntary nervous system, and it is involved in regulating the internal environment of the body. It carries signals from internal organs to the central nervous system, and from the central nervous system to the internal organs. In this way, it is involved in regulating things like digestion and heartbeat, which are generally outside the realm of conscious control. The autonomic nervous system can be further subdivided into sympathetic and parasympathetic nerve fibers. The sympathetic nervous system plays a large role in stimulating and mobilizing energy resources, while the parasympathetic nervous system acts to conserve energy. For example, if you're in a frightening situation, the sympathetic nervous system will cause your heart rate to increase, your blood pressure to increase, and your sweat glands to be stimulated. If you're eating a meal, however, and are not frightened, your parasympathetic nervous system will stimulate digestion, increase salivation, and slow your heart rate. Due to these functions, the sympathetic nervous system is often described as being involved in fight or flight responses, while the parasympathetic nervous system is described as being involved in rest and digest responses. Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the lobes of the brain and some major landmarks of the brain surface that can be seen from a lateral view. The brain is made up of two halves, known as cerebral hemispheres. When looking at the brain from a lateral view, which just means looking at it from the side, we will only see one cerebral hemisphere, but the landmarks I will point out are found on both cerebral hemispheres. The surface of the cerebral hemispheres is covered with a thick layer of brain tissue known as the cerebral cortex. The cerebral cortex is folded to create more surface area, forming these ridges, known as gyri, and grooves, known as sulci. This large sulcus is known as the central sulcus. It divides the frontal lobe from the parietal lobe. This sulcus is known as the Sylvian fissure, and it separates the temporal lobe from the rest of the cerebral hemisphere. The occipital lobe is found at the back of the brain, but it doesn't have such a clear division separating it from the rest of the brain. There are certain functions associated with the lobes, but any short description of these functions is oversimplified, as each is involved in many different processes. The frontal lobes are important for movement and higher-order cognition like rational thought, decision-making, and planning. This area in front of the central sulcus is known as the precentral gyrus, and it is essential for motor control as it is the location of the primary motor cortex. The parietal lobes are important for processing of sensory information and are involved in attention and our representation of the space around us. This area in the parietal lobe, the postcentral gyrus, contains the primary somatosensory cortex and is where the sense of touch is processed. The temporal lobe is involved in processing auditory signals and also is important in aspects of learning and memory. The occipital lobes contain major visual processing areas of the brain like the primary visual cortex.
Anytime someone hears the term motivation, they should really supplant that with dopamine. Because without dopamine, you don't have motivation.
All drugs of abuse cause this unnatural rise of dopamine. And because of that, they're among the most powerful experiences our brains can have. Individuals struggling with addiction are actually battling millions of years of evolution because our brains are exquisitely evolved to seek rewards.
Body decreases the production of dopamine. Eventually, you can't even get enough dopamine produced to get out of bed, let alone produce good relationships and good decision making.
Methadone is our gold standard. It's the most studied, it's been around forever. You attend a methadone clinic every day, you get a dose of medication. It fills your receptor sites so that the cravings don't happen.
So I included that one because overdosing on drugs is, the last I checked, is the number one cause of death in the United States for people under 50 years old. And it's yeah, it's a major problem, and a lot of those are opioids. Some of them prescription opioids, some of them illegal opioids. And yeah, as the video kind of explained, it these medications trigger the like reward centers, the like I enjoyed that and I would like to experience that again centers in your brain so strongly that it makes you really really want more, but it desensitizes your body so you need higher and higher levels, and the result of that is regular stuff that would trigger those. I enjoyed that. I would like to experience that again, whether it be a nice conversation or a meal, tasting your favorite food, all sorts of different things stop being enough. And so you put yourself in this position where nothing is enjoyable that used to be enjoyable. And the only thing that you find can give you this enjoyment is higher and higher doses of these different drugs, to the point where it eventually does something like it shuts down it like paralyzes the muscles that you need to breathe and you die of suffocation, stuff like that. So it is not something to be played with, and it is a very sad thing how many Americans die because of overdosing on stuff like that. And then moving on to the sensory system. Your textbook called these minor senses. I'm not quite sure I'd put taste and smell or even some of these different senses as minor, but I kind of get where they're coming from. But so senses of the skin, our skin has specific sensory cells for experiencing touch. And not just that, but there are like four different kinds of touch sensation or sensory touch neurons in your hands, for example. And like some are very very fine tuned and like they're on your fingertips, and they can do a really good job of feeling fine, small things. Others are more about feeling pressure. Others are more about feeling how the skin stretches and different things like that, and they all kind of serve a specific role of being able to feel texture versus being able to feel small things versus being able to handle tools and variety of things like that. But yeah, sensations for touch, for heat, for cold, for pressure, and for pain. And then your senses of taste and smell, and they're kind of related. The video mentions a little bit about that. So you have your taste buds; those are your receptors for taste. Video goes into that a little bit. Your olfactory receptors are your receptors for smell. And then there's something smell accommodation. Most of our senses, there's an accommodation that can happen where after a short period of stimulation, the receptors become insensitive to that specific stimulus, and a person does not smell it anymore or does not experience whatever sensation is. So think like if you sit at the edge of a swimming pool, when you first put your legs in, you can feel the water around your legs. You can feel like where the water level is hitting the side of your legs, or the outside of your legs. But then after just like a minute or two, you don't really feel the water anymore. That would be sensory accommodation. Or you walk into a room and there's a strong smell, but then after a couple minutes you can't really smell it anymore. That is also sensory accommodation. And our brains do that for a wide variety of our senses.
In 1995, the British Medical Journal published an astonishing report about a 29-year-old builder. He accidentally jumped onto a 15-centimeter nail, which pierced straight through his steel-toed boot. He was in such agonizing pain that even the smallest movement was unbearable. But when the doctors took off his boot, they faced a surprising sight. The nail had never touched his foot at all. For hundreds of years, scientists thought that pain was a direct response to damage. By that logic, the more severe an injury is, the more pain it should cause. But as we've learned more about the science of pain, we've discovered that pain and tissue damage don't always go hand in hand, even when the body's threat signaling mechanisms are fully functioning. We're capable of experiencing severe pain out of proportion to an actual injury, and even pain without any injury, like the builder, or the well-documented cases of male partners of pregnant women experiencing pain during the pregnancy or labor. What's going on here? There are actually two phenomena at play: the experience of pain and a biological process called nociception. Nociception is part of the nervous system's protective response to harmful or potentially harmful stimuli. Sensors in specialized nerve endings detect mechanical, thermal, and chemical threats. If enough sensors are activated, electrical signals shoot up the nerve to the spine and onto the brain. The brain weighs the importance of these signals and produces pain if it decides the body needs protection. Typically, pain helps the body avoid further injury or damage, but there are a whole set of factors besides nociception that can influence the experience of pain and make pain less useful. First, there are biological factors that amplify nociceptive signals to the brain. If nerve fibers are activated repeatedly, the brain may decide they need to be more sensitive to adequately protect the body from threats. More stress sensors can be added to nerve fibers until they become so sensitive that even light touches to the skin spark intense electrical signals. In other cases, nerves adapt to send signals more efficiently, amplifying the message. These forms of amplification are most common in people experiencing chronic pain, which is defined as pain lasting more than 3 months. When the nervous system is nudged into an ongoing state of high alert, pain can outlast physical injury. This creates a vicious cycle in which the longer pain persists, the more difficult it becomes to reverse. Psychological factors clearly play a role in pain too, potentially by influencing nociception and by influencing the brain directly. A person's emotional state, memories, beliefs about pain, and expectations about treatment can all influence how much pain they experience. In one study, children who reported believing that they had no control over pain actually experienced more intense pain than those who believed they had some control. Features of the environment matter too. In one experiment, volunteers with a cold rod placed on the back of their hand reported feeling more pain when they were shown a red light than a blue one, even though the rod was the same temperature each time. Finally, social factors like the availability of family support can affect perception of pain. All of this means that a multi-pronged approach to pain treatment that includes pain specialists, physical therapists, clinical psychologists, nurses, and other healthcare professionals is often most effective. We're only beginning to uncover the mechanisms behind the experience of pain, but there are some promising areas of research. Until recently, we thought the glial cells surrounding neurons were just support structures, but now we know they have a huge role in influencing nociception. Studies have shown that disabling certain brain circuits in the amygdala can eliminate pain in rats, and genetic testing in people with rare disorders that prevent them from feeling pain have pinpointed several other possible targets for drugs and perhaps eventually gene therapy. Ever wonder how pain relievers work or how other animals experience pain? Check out these lessons to learn more.
Some of that with like how strongly you experience the pain, and then also how you like with the kids about if they felt that they could control it, that they reported the pain as being less. A little bit of that relates to something called the placebo effect that maybe you're familiar with, but it is where if people think that they are receiving a medication that lessens their pain, oftentimes they say that their pain ends up being less than it was before. Or if they think they are receiving a medication to help fight a disease, they actually have a higher percentage of recovery, or they have a quicker recovery on average than people who don't think that they're receiving anything. And when groups do studies on, like, does this medication help cure a disease, they usually need three categories. There's a category that receives nothing, a category of people that receives a placebo, so they think they're getting the medicine, but they're not, and then a category of people that are actually getting the medication. And they need that middle category, that placebo category, because sometimes they think that a medication is working, but it's actually the recovery rate is the same as the placebo group's rate. And so, like, oh, this medication doesn't actually do anything. It's just the fact that people think that they're getting medicine is helping their bodies recover. There's just some crazy, not very well understood, but complicated and amazing aspects of how stuff like just thinking that you're getting medication for a disease can help you overcome that disease better and more quickly. And it's even stuff like they've done studies on the size of the pill that a person takes if they're taking a placebo pill, or if it's a red pill versus a blue pill versus a green pill, and that different sizes and different, or if it's like one large versus two small, and all of these different things end up having different outcomes with the placebo effect, because on average our brains perceive the benefit of the medication is going to be different depending on those different factors. And yeah, it's pretty crazy.
Why does your mouth feel like it's on fire when you eat a spicy pepper? And how do you soothe the burn? Why does wasabi make your eyes water? And how spicy is the spiciest spice? Let's back up a bit. First, what is spiciness? Even though we often say that something tastes spicy, it's not actually a taste, like sweet or salty or sour. Instead, what's really happening is that certain compounds in spicy foods activate the type of sensory neurons called polymodal nociceptors. You have these all over your body, including your mouth and nose, and they're the same receptors that are activated by extreme heat. So when you eat a chili pepper, your mouth feels like it's burning because your brain actually thinks it's burning. The opposite happens when you eat something with menthol in it. The cool, minty compound is activating your cold receptors. When these heat-sensitive receptors are activated, your body thinks it's in contact with a dangerous heat source and reacts accordingly. This is why you start to sweat and your heart starts beating faster. The peppers have elicited the same fight-or-flight response with which your body reacts to most threats. But you may have noticed that not all spicy foods are spicy in the same way, and the difference lies in the types of compounds involved. The capsaicin and piperine found in black pepper and chili peppers are made up of larger, heavier molecules called alkyl amides, and those mostly stay in your mouth. Mustard, horseradish, and wasabi are made up of smaller molecules called isothiocyanates that easily float up into your sinuses. This is why chili peppers burn your mouth and wasabi burns your nose. The standard measure of a food's spiciness is its rating on the Scoville scale, which measures how much its capsaicin content can be diluted before the heat is no longer detectable to humans. A sweet bell pepper gets 0 Scoville heat units, while Tabasco sauce clocks in between 1,200 and 2,400 units. The race to create the hottest pepper is a constant battle, but two peppers generally come out on top: the Trinidad Moruga Scorpion and the Carolina Reaper. These peppers measure between 1.5 and 2 million Scoville heat units, which is about half the units found in pepper spray. So why would anyone want to eat something that causes such high levels of pain? Nobody really knows when or why humans started eating hot peppers. Archaeologists have found spices like mustard along with human artifacts dating as far back as 23,000 years ago, but they don't know whether the spices were used for food or medication or just decoration. More recently, a 6,000-year-old crockpot lined with charred fish and meat also contained mustard. One theory says that humans started adding spices to food to kill off bacteria, and some studies show that spice developed mostly in warmer climates, where microbes also happen to be more prevalent. But why we continue to subject ourselves to spicy food today is still a bit of a mystery. For some people, eating spicy food is like riding roller coasters. They enjoy the ensuing thrill, even if the immediate sensation is unpleasant. Some studies have even shown that those who like to eat hot stuff are more likely to enjoy other adrenaline-rich activities like gambling. The taste for spicy food may even be genetic. And if you're thinking about training a bit to up your tolerance for spice, know this. According to some studies, the pain doesn't get any better. You just get tougher. In fact, researchers have found that people who like to eat spicy foods don't rate the burn any less painful than those who don't. They just seem to like the pain more. So torment your heat receptors all you want, but remember, when it comes to spicy food, you're going to get burned. It's the first sense you use when you're born. 1 out of every 50 of your genes is dedicated to it. It must be important, right? Okay, take a deep breath through your nose. It's your sense of smell, and it's breathtakingly powerful. As an adult, you can distinguish about 10,000 different smells. Here's how your nose does it. Smell starts when you sniff molecules from the air into your nostrils. 95% of your nasal cavity is used just to filter that air before it hits your lungs. But at the very back of your nose is a region called the olfactory epithelium, a little patch of skin that's key to everything you smell. The olfactory epithelium has a layer of olfactory receptor cells, special neurons that sense smells, like the taste buds of your nose. When odor molecules hit the back of your nose, they get stuck in a layer of mucus covering the olfactory epithelium. As they dissolve, they bind to the olfactory receptor cells, which fire and send signals through the olfactory tract up to your brain. As a side note, you can tell a lot about how good an animal's sense of smell is by the size of its olfactory epithelium. A dog's olfactory epithelium is 20 times bigger than your puny human one. But there's still a lot we don't know about this little patch of cells, too. For example, our olfactory epithelium is pigmented, and scientists don't really know why. But how do you actually tell the difference between smells? It turns out that your brain has 40 million different olfactory receptor neurons. So odor A might trigger neurons 3, 427, and 988, and odor B might trigger neurons 8, 76, and 2,496,678. All of these different combinations let you detect a staggeringly broad array of smells. Plus, your olfactory neurons are always fresh and ready for action. They're the only neuron in the body that gets replaced regularly, every 4 to 8 weeks. Once those neurons are triggered, the signal travels through a bundle called the olfactory tract to destinations all over your brain, making stops in the amygdala, the thalamus, and the neocortex. This is different from how sight and sound are processed. Each of those signals goes first to a relay center in the middle of the cerebral hemisphere, and then out to other regions of the brain. But smell, because it evolved before most of your other senses, takes a direct route to these different regions of the brain, where it can trigger your fight or flight response, help you recall memories, or make your mouth water. But even though we've all got the same physiological setup, 2 nostrils and millions of olfactory neurons, not everybody smells the same things. One of the most famous examples of this is the ability to smell so-called asparagus pea. For about a quarter of the population, urinating after eating asparagus means smelling a distinct odor. The other 75% of us don't notice. And this isn't the only case of smells differing from nose to nose. For some people, the chemical androstenone smells like vanilla. To others, it smells like sweaty urine. Which is unfortunate because androstenone is commonly found in tasty things like pork. So with the sweaty urine smellers in mind, pork producers will castrate male pigs to stop them from making androstenone. The inability to smell a scent is called anosmia, and there are about 100 known examples. People with allicin anosmia can't smell garlic. Those with eugenol anosmia can't smell cloves. And some people can't smell anything at all. This kind of full anosmia can have several causes. Some people are born without a sense of smell. Others lose it after an accident or during an illness. If the olfactory epithelium gets swollen or infected, it can hamper your sense of smell, something you might have experienced when you were sick. And not being able to smell anything can mess with your other senses too. Many people who can't smell at all also can't really taste the same way the rest of us do. It turns out that how something tastes is closely related to how it smells. As you chew your food, air is pushed up your nasal passage, carrying with it the smell of your food. Those scents hit your olfactory epithelium and tell your brain a lot about what you're eating. Without the ability to smell, you lose the ability to taste anything more complicated than the 5 tastes your taste buds can detect: sweet, salty, bitter, sour, and savory. So the next time you smell exhaust fumes, salty sea air, or roast chicken, you'll know exactly how you've done it, and perhaps be a little more thankful that you can.
All right. and then wrapping things up, a little bit of a look at our sense of hearing and then our sense of sight. So, when we look at the ear, there are three parts of the ear that we talk about. There's the outer ear, the middle ear, and the inner ear. So the outer ear is made up of the auricle, which is the outer flap, and its main purpose is to collect the sound waves, help direct them to the external auditory canal. This is the tube which goes into the head and ends at the tympanic membrane, the eardrum. That's the eardrum right there. So that's the outer ear, the auricle, and then the ear canal. Then the middle ear, it has a moist air-filled chamber containing three bones. These three bones are sometimes also called ossicles, and it's the malleus, the incus, and the stapes, which is Latin for the hammer, the anvil, and the stirrup. So that's the malleus there, the hammer, and then the anvil, and then the stirrup, and they're called that because the shape of them roughly resembles those objects. And then there's the oval window. That would be right here. That is a membrane-covered opening to the inner ear. And it's also kind of right here at the boundary between the middle ear and the inner ear. You have your eustachian tube that goes down to the back of your throat, basically, and that's what helps regulate the air pressure regulate the air pressure behind the eardrum and out of the eardrum, in front of the eardrum. And the inner ear. And so this has a bony labyrinth enclosing a membranous labyrinth. These are those labyrinths here. And there's the cochlea, which is a snail-shaped division of the inner ear labyrinth. So the cochlea there, the video will explain a little bit. And then there's also these semicircular canals, and these are mainly related to your sense of balance. So it's how you can tell if your head is tilted to the side, or if your head is tilted forward, or if you're upside down, even if you're not moving. That would be your static equilibrium. You're not moving, but you can tell you're not upright. And your dynamic equilibrium would be being able to identify senses of motion. And there's something that is kind of fun that you can do, though you have to be careful to catch the person in case they hurl themselves, if they fling their body. But you can take like a swivel chair and have a person sit on it, like facing backwards. So it's like their legs are kind of tucked up under them, and they are resting their head on its side on the back of the chair. It's kind of hard to explain, and then you can rotate the person so that they are facing the direction that they're rotating, and you can rotate them either forward or backward. And what happens was as they're rotating, there's fluid in these canals, these semicircular canals, that the fluid, when you first start to move, your body moves more than the fluid, and that triggers the sense of, oh, I'm in motion. But if you keep spinning the person, the fluid eventually ends up rotating at the same rate that your head is, and at that point they kind of feel like, oh, I don't really feel like I'm moving anymore. But if you stop them and you abruptly have the person look like straight ahead, like lift their head up and look straight ahead, the movement in the fluid in the semicircular canals is still rotating inside of those canals, and it makes them feel like they are moving now, even though they're actually standing still. And usually, like, they can't help but move their eyes up and down to kind of try to counteract the movement that they think that they feel. And sometimes, depending if you do it forward or backwards, a person can end up flinging their body forwards, and it almost feels like it's hard to keep your head up off of the floor, because it just, with how your body's reacting to the sense of movement, even though you're not moving anymore. So it's kind of a brain trick that you can play on yourself like that. All right, so a little bit about the sense of hearing.
Have you ever wondered how sounds make their way from the source all the way to your brain? Take a trumpet, for instance. When it's played, it makes sound waves in the air. The outer ear catches the waves, which then travel through a narrow passageway called the ear canal. The sound waves reach the eardrum, which is a membrane roughly half the size of a dime. They make the eardrum vibrate, which in turn vibrates three tiny bones called the malleus, incus, and stapes. These bones amplify or increase the sound vibrations and send them to the cochlea. The cochlea is shaped like a snail and is the size of a garden pea. It is filled with fluid, and the sound vibrations make this fluid ripple, which creates waves. Hair-like structures called stereocilia sit on top of hair cells and are grouped together as hair cell bundles inside the cochlea. The hair cells inside the cochlea ride these waves, and the hair bundles are moved. The hair bundle on top of the hair cell turns these movements into electrical signals. As the hair bundles are moved, ions rush into the top of the hair cells, causing the release of chemicals at the bottom of the hair cells. The chemicals bind to the auditory nerve cells and create an electrical signal, which travels along the auditory nerve to the brain. Different hair cells respond to different frequencies of sound. The hair cells at the base of the cochlea detect higher-pitched sounds, such as a piccolo or flute. The hair cells toward the top of the spiral detect progressively lower-pitched sounds, such as a trumpet or trombone. At the very top or apex of the spiral, the hair cells detect the lowest-pitched sounds, such as a tuba. The auditory nerve carries the electrical signal to the brain, which interprets the messages as sounds that we recognize and understand.
And finally, the sense of sight. So the eye, the main parts of the eye, there's a sclera, which is shaded blue here, but it's the white outer covering of the eye. And then the cornea, this is the transparent part that is what you're able to see through at the front of your eye. And then the choroid, this is a fragile thin layer with many blood vessels for nourishing the retina. The choroid is the red along in here, goes up till there, which is where the iris is. So the iris here contains muscles and is a colored part of the eye. So it's what makes your eyes brown or green or hazel, and then blue. And then there's the pupil. The pupil there is the circular opening in the iris, and it's what closes or dilates to control how much light we let into our eyes to adjust for brightness versus darkness. It's the equivalent of the it's related, it's similar to the aperture, the f-stop of a camera. And let's see, so it's pupil, and then the retina. The retina is what's shown here in yellow. That is we mainly think of the retina as the back part of the eye. It's composed of thousands of specialized neurons in their fibers. The two main kinds of sensory neurons in the retina are rods and cones. Rods are good at detecting light and movement. Cones good at detecting color. We have cones mainly in the middle, and so it's mainly things that is toward our center of our field of vision that we have good color perception. And then the rods are mainly toward the outside, so it's things in our peripheral vision that are better at detecting light and better at detecting motion. That's how that's laid out. And then the optic nerve here is where all the different neurons converge, where the nerve goes to the brain. You have a blind spot right there. There's some interesting things you can get that like it's like, look at these two dots, and then the little crosshairs will disappear because it puts the crosshairs in your blind spot and you can't see it if you're looking at these dots, kind of a thing. Then there's a lens that's shown here. It's a biconvex lens, also known in physics as a converging lens, and it focuses the light rays on the retina. So the light rays come in and it focuses, if there's an object right here that you're looking at, focuses it so that you get a clear image. That's something more of a physics topic that we get into in physics. Then you have the ciliary muscles and suspensory ligaments. You have the suspensory ligaments mentioned here. They help support and reshape the lens for the sake of getting stuff in focus. Then there's the aqueous humor. It is a transparent watery fluid between the cornea and the lens. That's this stuff in here. And then the vitreous humor is a clear, permanent, jelly-like substance in the posterior chamber of the eye. So there you go. And then one more video, and that does it for this overall chapter.
Sight, a precious gift we can't take for granted. Our eyes are windows to the wonders of the world. All we know and love, experience and discover, ponder and cherish. Let's peer into the workings of the eye, a unique and self-sustaining system. The reflected light of the world enters through the crystal transparency of the cornea, aqueous humor, lens, and vitreous humor to project onto the photoreceptors of the retina, whose impulses converge on the optic nerve, and then to the brain to be transfigured into imagery and imbued with meaning. Continual adjustments to the pupil and lens regulate the entry and focusing of light. In today's world, the eye's surface is constantly challenged to protect itself and adjust to changing conditions. Every blink helps our eye's natural defense system, the tear film, retain moisture and maintain visual acuity. Tears contain potent natural disinfectants to keep bacteria at bay. And the tear system washes away impurities to maintain eye health. The miraculous biology of our eyes inspires us to develop ways we can protect and maintain comfort for these remarkable structures. For all the wonders our eyes provide throughout our lives, we owe them our attention and ongoing care.
And that wraps up our new content for the year for biology. So I'll get a study guide to you really soon for what to study for your little human anatomy test tomorrow, or sorry, not tomorrow, next week. And yeah, so between the foundational stuff of how the cell works, of how genes work, to the overview of the different living organism kingdoms, bacteria, all the way up to animals, to then this little bit of human anatomy. Hopefully, you learn some interesting stuff. Hopefully, you learn some stuff that changes how you view the world or how you understand and interact with the world. And yep, yeah, I think it's a fascinating subject, and there's always more than I can learn about it because it's so vast, even though it's what I majored in for my undergrad degree. But yep, hope you have a great summer. And at least for most of you, I will see you next year.